Macrophages, pronunciation, /µkrə, feɪdʒ/Greek, big eaters, from Greek mu alpha kappa rho, macros, equals large, phi alpha gamma epsilon nu, phageín, equals to eat, are a type of white blood cell that engulfs and digests cellular debris, foreign substances, microbes, cancer cells, and anything else that does not have the types of proteins specific to healthy body cells on its surface in a process called phagocytosis. These large phagocytes are found in essentially all tissues, where they patrol for potential pathogens by amoeboid movement. They take various forms, with various names, throughout the body, e.g., histiocytes, Kupffer cells, alveolar macrophages, microglia, and others, but all are part of the mononuclear phagocyte system. Besides phagocytosis, they play a critical role in nonspecific defense, innate immunity, and also help initiate specific defense mechanisms, adaptive immunity, by recruiting other immune cells such as lymphocytes. For example, they are important as antigen presenters to T cells. In humans, dysfunctional macrophages cause severe diseases such as chronic granulomatous disease that result in frequent infections. Beyond increasing inflammation and stimulating the immune system, macrophages also play an important anti-inflammatory role and can decrease immune reactions through the release of cytokines. Macrophages that encourage inflammation are called M1 macrophages, whereas those that decrease inflammation and encourage tissue repair are called M2 macrophages. This difference is reflected in their metabolism. M1 macrophages have the unique ability to metabolize arginine to the killer molecule nitric oxide, whereas rodent M2 macrophages have the unique ability to metabolize arginine to the repair molecule ornithine. However, this dichotomy has been recently questioned as further complexity has been discovered. Human macrophages are about 21 micrometers, 0.00083 in diameter and are produced by the differentiation of monocytes in tissues. They can be identified using flow cytometry or immunohistochemical staining by their specific expression of proteins such as CD14, CD40, CD11b, CD64, F4/80, mice/EMR1, human, lysozyme M, MAC1/MAC3 and CD68. Macrophages were first discovered by Elie Mechnikov, a Russian zoologist, in 1884. Life cycle. When a monocyte enters damaged tissue through the endothelium of a blood vessel, a process known as the leukocyte extravasation, it undergoes a series of changes to become a macrophage. Monocytes are attracted to a damaged site by chemical substances through chemotaxis, triggered by a range of stimuli including damaged cells, pathogens, and cytokines released by macrophages already at the site. At some sites such as the testis, macrophages have been shown to populate the organ through proliferation. Unlike short-lived neutrophils, macrophages survive longer in the body, up to several months. Function. Phagocytosis. Macrophages are professional phagocytes and are highly specialized in removal of dying or dead cells and cellular debris. This role is important in chronic inflammation, as the early stages of inflammation are dominated by neutrophils, which are ingested by macrophages if they come of age. See CD31 for a description of this process. The neutrophils are at first attracted to a site, where they proliferate, before they are phagocytized by the macrophages. When at the site, the first wave of neutrophils, after the process of aging and after the first 48 hours, stimulate the appearance of the macrophages, whereby these macrophages will then ingest the aged neutrophils. The removal of dying cells is, to a greater extent, handled by fixed macrophages, which will stay at strategic locations such as the lungs, liver, neural tissue, bone, spleen, and connective tissue, ingesting foreign materials such as pathogens and recruiting additional macrophages if needed. When a macrophage ingests a pathogen, the pathogen becomes trapped in a phagosome, which then fuses with a lysosome. Within the phagolysosome, enzymes and toxic peroxides digest the pathogen. However, some bacteria, such as Mycobacterium tuberculosis, have become resistant to these methods of digestion. Typhoidal salmonella induce their own phagocytosis by host macrophages in vivo, and inhibit digestion by lysosomal action, thereby using macrophages for their own replication and causing macrophage apoptosis. Macrophages can digest more than 100 bacteria before they finally die due to their own digestive compounds. Role in adaptive immunity. Macrophages are versatile cells that play many roles. As scavengers, they rid the body of worn-out cells and other debris. Along with dendritic cells, they are foremost among the cells that present antigens, a crucial role in initiating an immune response. As secretory cells, monocytes and macrophages are vital to the regulation of immune responses and the development of inflammation, they produce a wide array of powerful chemical substances, monokines, including enzymes, complement proteins, and regulatory factors such as interleukin-1. At the same time, they carry receptors for lymphokines that allow them to be activated into single-minded pursuit of microbes and tumor cells. After digesting a pathogen, a macrophage will present the antigen, a molecule, most often a protein found on the surface of the pathogen and used by the immune system for identification of the pathogen to the corresponding helper T cell. The presentation is done by integrating it into the cell membrane and displaying it attached to an MHC class II molecule, MindTap, indicating to other white blood cells that the macrophage is not a pathogen, despite having antigens on its surface. Eventually, the antigen presentation results in the production of antibodies that attach to the antigens of pathogens, making them easier for macrophages to adhere to with their cell membrane and phagocytose. In some cases, pathogens are very resistant to adhesion by the macrophages. The antigen presentation on the surface of infected macrophages, in the context of MHC class II, in a lymph node stimulates Th1, type 1 helper T cells, to proliferate, mainly due to IL-12 secretion from the macrophage. When a B cell in the lymph node recognizes the same unprocessed surface antigen on the bacterium with its surface-bound antibody, the antigen is endocytosed and processed. The processed antigen is then presented in MindTap on the surface of the B cell. T cells that express the T cell receptor which recognizes the antigen MindTap complex, with co-stimulatory factors CD40 and CD40L, cause the B cell to produce antibodies that help opsonization of the antigens so that the bacteria can be better cleared by phagocytes. Macrophages provide yet another line of defense against tumor cells and somatic cells infected with fungus or parasites. Once a T cell has recognized its particular antigen on the surface of an aberrant cell, the T cell becomes an activated effector cell, producing chemical mediators known as lymphokines that stimulate macrophages into a more aggressive form. Macrophage subtypes. There are several activated forms of macrophages. In spite of a spectrum of ways to activate macrophages, there are two main groups designated M1 and M2. M1 macrophages, as mentioned earlier, previously referred to as classically activated macrophages, M1 killer macrophages are activated by LPS and IFN gamma, and secrete high levels of IL-12 and low levels of IL-10. In contrast, the M2 repair designation, also referred to as alternatively activated macrophages, broadly refers to macrophages that function in constructive processes like wound healing and tissue repair, and those that turn off damaging immune system activation by producing anti-inflammatory cytokines like IL-10. M2 is the phenotype of resident tissue macrophages, and can be further elevated by IL-4. M2 macrophages produce high levels of IL-10, TGF-beta and low levels of IL-12. Tumor-associated macrophages are mainly of the M2 phenotype, and seem to actively promote tumor growth. Both M1 and M2 macrophages play a role in promotion of atherosclerosis. M1 macrophages promote atherosclerosis by inflammation. M2 macrophages can remove cholesterol from blood vessels, but when the cholesterol is oxidized, the M2 macrophages become apoptotic foam cells contributing to the atheromatous plaque of atherosclerosis. Role in muscle regeneration. The first step to understanding the importance of macrophages in muscle repair, growth, and regeneration is that there are two waves of macrophages with the onset of damageable muscle use subpopulations that do and do not directly have an influence on repairing muscle. The initial wave is a phagocytic population that comes along during periods of increased muscle use that are sufficient to cause muscle membrane lysis and membrane inflammation, which can enter and degrade the contents of injured muscle fibers. These early invading, phagocytic macrophages reach their highest concentration about 24 hours following the onset of some form of muscle cell injury or reloading. Their concentration rapidly declines after 48 hours. The second group is the non-phagocytic types that are distributed near regenerative fibers. These peak between 2 and 4 days and remain elevated for several days during the hopeful muscle rebuilding. The first subpopulation has no direct benefit to repairing muscle, while the second non-phagocytic group does. It is thought that macrophages release soluble substances that influence the proliferation, differentiation, growth, repair, and regeneration of muscle, but at this time the factor that is produced to mediate these effects is unknown. It is known that macrophages' involvement in promoting tissue repair is not muscle-specific. They accumulate in numerous tissues during the healing process phase following injury. Role in wound healing. Macrophages are essential for wound healing. They replace polymorphonuclear neutrophils as the predominant cells in the wound by day 2 after injury. Attracted to the wound site by growth factors released by platelets and other cells, monocytes from the bloodstream enter the area through blood vessel walls. Numbers of monocytes in the wound peak 1 to 1.5 days after the injury occurs. Once they are in the wound site, monocytes mature into macrophages. The spleen contains half the body's monocytes in reserve ready to be deployed to injured tissue. The macrophage's main role is to phagocytize bacteria and damaged tissue, and they also debride damaged tissue by releasing proteases. Macrophages also secrete a number of factors such as growth factors and other cytokines, especially during the 3rd and 4th post-wound days. These factors attract cells involved in the proliferation stage of healing to the area. Macrophages may also restrain the contraction phase. Macrophages are stimulated by the low oxygen content of their surroundings to produce factors that induce and speed angiogenesis, and they also stimulate cells that reepithelialize the wound, create granulation tissue, and lay down a new extracellular matrix. By secreting these factors, macrophages contribute to pushing the wound healing process into the next phase. Role in limb regeneration. Scientists have elucidated that as well as eating up material debris, macrophages are involved in the typical limb regeneration in the salamander. They found that removing the macrophages from a salamander resulted in failure of limb regeneration and a scarring response. Role in iron homeostasis. Main article: Human iron metabolism. As described above, macrophages play a key role in removing dying or dead cells and cellular debris. Erythrocytes have a lifespan on average of 120 days and so are constantly being destroyed by macrophages in the spleen and liver. Macrophages will also engulf macromolecules, and so play a key role in the pharmacokinetics of parenteral irons. The iron that is released from the hemoglobin is either stored internally in ferritin or is released into the circulation via ferroportin. In cases where systemic iron levels are raised, or where inflammation is present, raised levels of hepcidin act on macrophage ferroportin channels, leading to iron remaining within the macrophages. Tissue macrophages. A majority of macrophages are stationed at strategic points where microbial invasion or accumulation of foreign particles is likely to occur. These cells together as a group are known as the mononuclear phagocyte system and were previously known as the reticuloendothelial system. Each type of macrophage, determined by its location, has a specific name. Cell name: anatomical location. Adipose tissue macrophages: adipose tissue. Monocytes: bone marrow/blood. Kupffer cells: liver. Sinus histiocytes: lymph nodes. Alveolar macrophages: dust cells, pulmonary alveoli of lungs. Tissue macrophages: histiocytes, leading to giant cells, connective tissue. Langerhans cells: skin and mucosa. Microglia: central nervous system. Hofbauer cells: placenta. Intraglomerular mesangial cells: kidney. Osteoclasts: bone. Epithelioid cells: granulomas. Red pulp macrophages: sinusoidal lining cells, red pulp of spleen. Peritoneal macrophages: peritoneal cavity. Lysomac Peyer's patch. Investigations concerning Kupffer cells are hampered because in humans, Kupffer cells are only accessible for immunohistochemical analysis from biopsies or autopsies. From rats and mice, they are difficult to isolate, and after purification, only approximately 5 million cells can be obtained from one mouse. Macrophages can express paracrine functions within organs that are specific to the function of that organ. In the testis, for example, macrophages have been shown to be able to interact with Leydig cells by secreting 25-hydroxycholesterol, an oxysterol that can be converted to testosterone by neighboring Leydig cells. Also, testicular macrophages may participate in creating an immune-privileged environment in the testis, and in mediating infertility during inflammation of the testis. Cardiac-resident macrophages participate in electrical conduction via gap junction communication with cardiac myocytes. Macrophages can be classified on basis of the fundamental function and activation. According to this grouping, there are classically activated macrophages, wound-healing macrophages, alternatively activated macrophages, and regulatory macrophages, regs. Disease. Due to their role in phagocytosis, macrophages are involved in many diseases of the immune system. For example, they participate in the formation of granulomas, inflammatory lesions that may be caused by a large number of diseases. Some disorders, mostly rare, of ineffective phagocytosis and macrophage function have been described, for example, as a host for intracellular pathogens. In their role as a phagocytic immune cell, macrophages are responsible for engulfing pathogens to destroy them. Some pathogens subvert this process and instead live inside the macrophage. This provides an environment in which the pathogen is hidden from the immune system and allows it to replicate. Diseases with this type of behavior include tuberculosis, caused by Mycobacterium tuberculosis, and leishmaniasis, caused by Leishmania species. Tuberculosis. Once engulfed by a macrophage, the causative agent of tuberculosis, Mycobacterium tuberculosis, avoids cellular defenses and uses the cell to replicate. Leishmaniasis. Upon phagocytosis by a macrophage, the Leishmania parasite finds itself in a phagocytic vacuole. Under normal circumstances, this phagocytic vacuole would develop into a lysosome and its contents would be digested. Leishmania alter this process and avoid being destroyed. Instead, they make a home inside the vacuole. Chikungunya. Infection of macrophages in joints is associated with local inflammation during and after the acute phase of chikungunya, caused by CHIKV or chikungunya virus. Others. Adenovirus, most common cause of pink eye, can remain latent in a host macrophage, with continued viral shedding 6–18 months after initial infection. Brucella species can remain latent in a macrophage via inhibition of phagosome-lysosome fusion, causes brucellosis, undulant fever. Legionella pneumophila, the causative agent of Legionnaire's disease, also establishes residence within macrophages. Heart disease. Macrophages are the predominant cells involved in creating the progressive plaque lesions of atherosclerosis. Focal recruitment of macrophages occurs after the onset of acute myocardial infarction. These macrophages function to remove debris, apoptotic cells, and to prepare for tissue regeneration. HIV infection. Macrophages also play a role in human immunodeficiency virus, HIV, infection. Like T cells, macrophages can be infected with HIV, and even become a reservoir of ongoing virus replication throughout the body. HIV can enter the macrophage through binding of GP120 to CD4 and second membrane receptor, CCR5, a chemokine receptor. Both circulating monocytes and macrophages serve as a reservoir for the virus. Macrophages are better able to resist infection by HIV-1 than CD4 plus T cells, although susceptibility to HIV infection differs among macrophage subtypes. Cancer. Macrophages contribute to tumor growth and progression. Attracted to oxygen-starved, hypoxic, and necrotic tumor cells, they promote chronic inflammation. Inflammatory compounds such as tumor necrosis factor, TNF, alpha released by the macrophages activate the gene switch nuclear factor kappa B. NF-kappa B then enters the nucleus of a tumor cell and turns on production of proteins that stop apoptosis and promote cell proliferation and inflammation. Moreover, macrophages serve as a source for many pro-angiogenic factors including vascular endothelial factor, VEGF, tumor necrosis factor alpha, TNF-alpha, macrophage colony stimulating factor, MCSF/CSF1, and IL-1 and IL-6 contributing further to the tumor growth. Macrophages have been shown to infiltrate a number of tumors. Their number correlates with poor prognosis in certain cancers including cancers of breast, cervix, bladder, and brain. Tumor-associated macrophages, TAMs, are thought to acquire an M2 phenotype, contributing to tumor growth and progression. Research in various study models suggests that the cooperation of T cells and macrophages is important to suppress tumors. This cooperation involves not only the direct contact of T cell and macrophage, with antigen presentation, but also includes the secretion of adequate combinations of cytokines, which enhance T cell anti-tumor activity. Recent study findings suggest that by forcing IFN-alpha expression in tumor-infiltrating macrophages, it is possible to blunt their innate protumoral activity and reprogram the tumor microenvironment toward more effective dendritic cell activation and immune effector cell cytotoxicity. Obesity. Increased number of pro-inflammatory macrophages within obese adipose tissue contributes to obesity complications including insulin resistance and diabetes type 2. Within the fat, adipose, tissue of CCR2 deficient mice, there is an increased number of eosinophils, greater alternative macrophage activation, and a propensity towards type 2 cytokine expression. Furthermore, this effect was exaggerated when the mice became obese from a high-fat diet.
Please subscribe and thanks for watching.